Allergen-specific immunotherapy is a curative therapy that is available for the major allergens, and also different adjuvants are currently already on the market. But it's not so clear which adjuvant is the best one and how they actually can promote the success of the therapy. In this presentation, I would like to summarize some data and some immunological knowledge around the immunotherapy. This is my conflicts of interest which I would like to display. I've been working together with several companies and received research funding from DFG and BMBF for this kind of research. I would also like to highlight one of the reviews I've written together with several colleagues, which occurred in 2017. Now, for allergen-specific immunotherapy, alum is kind of the gold standard. It has been used as adjuvant for many decades, not only in allergen-specific immunotherapy, but also in many vaccination designs. So it's kind of the ketchup among the spices on the market of adjuvants, but there are many more. So for example, shark oil has been used as an adjuvant, but also ceramide spermidine, which is called Vaxizone. There is a saponin, so a detergent, which has been used as adjuvant, but also chitin components were utilized, and many others, which I've summarized here in this table. I don't want to go through all details, but I think it's interesting to note that alum is on the market since 1930s already, and it activates the inflammasome that has been clear and has been investigated in a greater detail about a decade ago, which actually also very much highlighted the relevance of the inflammasome in general. The MF59 is available since 1997. It's an oil, squalene, which is actually at the end activating ATP and therefore has a stimulating effect on antigen presentation. Monophospholipid A is a derivative of a bacterial endotoxin and is used since 2009 and is the first and only toll-like receptor ligand approved in human vaccination. So it's kind of maybe very clear defined adjuvant really that is available. The other ones are investigated adjuvants. So these are, to my knowledge, not as widely used at the moment as the adjuvants I've shown you before, but saponin is one of them, which is investigated under the product number QS21. There is also an LIK hybrid technology approach from a company. It's called Antigen Express Incorporation, and it actually enhances MHC binding, so this could be also quite a different approach compared to the other one. Respimod is TLR7 and 8 agonist and has been also investigated in terms of adjuvant activity. And then also IMO 2099, which is an agonist for toll-like receptors 3, 7, 8, or 9. But there's the list continues, as you see. There's also the GLA, which is a TLR4 agonist. Then a lot of liposomes have also been used in different formulations. Particularly also the cationic lipid DNA complexes. Yeah, I mean, this is now also used in gene therapy, and then also the Vaxizone, which is a synthetic polycationic sphingolipid, derived from the erythroceramide. So this is another example, which can be assembled into liposomes and used as well. Then there's also the possibility to fuse antigens together with specific receptor or even antibodies to directly target a certain molecule to a specific cell. So this is called S-target and has been also used in allergy actually to target house dust mite. And there are others also like viral particles which has been used to mimic like a viral response. So all these adjuvants, enhancers, potentiators or depots follow kind of different philosophies and target different mechanisms in the immune system. I think this diverse list already makes clear that adjuvant is not equaling adjuvant. I think we probably have to distinguish a little bit. And I would like to summarize on a very simple way what we think about adjuvants and what they are doing. So if we inject the antigen in a subcutaneous approach, what the thought is happening is upon injection, we are actually getting, I'll activate the pointer. So what we think is that the antigen here at this stage is taken up by dendritic cells. It's actually transported into the lymph node, and in the lymph node, the antigen presenting cells is now activating so far uncommitted cells or already existing mature T cells. If they are not differentiated, they either differentiate into Th2, into Th17, Th1 cells, but maybe also into regulatory cells, or here the Th22 cells, which is the novel cell type which we discovered a couple of years ago. And these cells have a very different functionality, and I think this is very important to memorize, because now, of course, what we would like to see is that the adjuvant is driving the immune response of one of these cell types, but not all of them. And one of the things which we probably don't want is the activation of Th2 cells, which are actually giving rise to IgE production, which makes allergy even worse. No, what we really want are these guys here, regulatory T cells, or cells which are anti-inflammatory, or maybe also Th1 cells, which do have other functions but usually do not promote IgE production and thus allergic inflammation. So if you now look at the definition of adjuvants, what we are actually thinking of is to boost the antigen presentation here at this early level, where we are. Where we are. I have to activate. Sorry for this. Where we want to maximize the amount of allergen that actually maximally is presented here in the lymph node to the T cells. So this is what we really want. But I think the other attractive option, and maybe we can find an adjuvant which is not only enhancing the antigen presentation, but also modulates the differentiation of naive T cells to one of these subtypes and not all of them. The adjuvant effect is important because the more antigen we inject, the higher is the risk that we have side effects also by the antigens. For example, in allergen-specific immunotherapy, you don't want to trigger anaphylaxis, and you also don't want to see differentiation of too many Th2 cells. So here, I think the adjuvant may serve to reduce the amount of allergen and thereby reduce side effects. And I think for the companies it's also obvious an effect that they don't need to spend too much money on putting too much antigen into the drugs. The The other thing I would like to highlight, also important of course, is what's happening to the immunoglobulins. So, and this is in fact one of the aspects it becomes apparent here, that the classical functions of adjuvants was always the immunoglobulin production. So if we think of viral vaccinations, the first thing you would look at is at the immunoglobulin, so-called zero conversion from IgM to IgG. That really matters, and the better this is, the better the adjuvant would be. So if we now look, this is a very confusing slide, but I think also very impressive if you look at the naive T cells, and then we have these T cell phenotypes, Th1 cells, Th22, Th2 cells, Th9, Th17, and regulatory T cells. And we think that all of them are coming from this naive precursor cell, and it becomes immediately apparent, and this is really an area which excites me personally very much. Yeah, if we could influence this process here initially, yeah, we get all these kind of cytokines released by these T cells, so interferon gamma, IL-22 here by the Th22 cells, IL-4, IL-5, IL-13, IL-31, IL-3 from the Th2 cells, IL-9 by the Th9 cells, Th17 cells, mainly characterized by IL-17, and the regulatory cytokines IL-10 and TGF-beta by the regulatory T cells. And these cytokines have now distinct effects on the target cells. And you see the target cells are quite diverse. This ranges from dendritic cells, but also structural cells here in the case of interferon gamma. Th22 cells, interesting, yeah, they have a more restricted profile to epithelial cells, so they are really affecting the mesenchymal branch of the tissue. And then the Th2 cells, B cells, eosinophils of course, but they also have effects on epithelial cells, keratinocytes. In fact, we recently could show that type 2 cytokines are driving epithelial cells towards a type 2 phenotype. And then the Th9 cells important for mast cells, IL-17 important for epithelial cells, and now these cells are mediating functions and symptoms at the end of the day. Yeah? And this is listed here at the bottom level, so specific immunity of course by MHC, but for example interferon gamma induces apoptosis in keratinocytes and generates a display of sponchiosis. If you look at immunohistochemistry pictures also of artificial skin culture, so it really punches hole into it, whereas the type 2 cells give rise to the bronchial hyperreactivity, hypersensitivity, airway remodeling, some cytokines promote itch, etc. So really these cytokines are then at the end mediating clinical symptoms, not directly in most cases, an indirect effect by these cells here in the middle. So these cytokines here at the beginning are critical to drive these different directions. Yeah? And now it really gets interesting because, of course, we would like to use the adjuvants and the immune modulators specifically in this area to change the phenotype of the T cells. And of course, it would be most preferable, if you could go along the Treg way here, but we don't really know this signals which are going along that way. In fact, we think that this pathway may be the kind of default pathway, and this is illustrated here in this scheme. This is an hypothesis, really. It has not been proven, really, and it will be difficult to prove this in an in vivo situation, really. But the observation is, if you activate a naive T cell, which has not seen an antigen before, what you always see is the induction of a transcription factor which is called FOXP3. It's kind of an activation marker. Whenever you activate the T cell, you get along this transcription factor. And this, in fact, is essential for the induction of regulatory cells which promote tolerance. And we hypothesized that any signal which diverts this default pathway away from the standard kind of commitment, such as IL-4. Yeah? IL-4 is inducing GATA3 and then generates either Th2 or together with TGF-beta the Th9 phenotype. Or here in the other direction, yeah, T-bet interferon gamma, yeah, you need IL-12 to direct away these T cells from the path of tolerance into inflammation. So this is the kind of concept. And if you convert now this concept into adjuvants and immunomodulators, yeah, it becomes apparent that one of the things we may want to do would be to neutralize the commitment towards the Th2 cells. And this is in fact something which we did in a clinical trial. But I will come later to this talk. But I think this is one of the exciting future perspectives. But before we come to the future aspects, we have to look at the current adjuvants. And here I think the dendritic cell is really, of course, in the focus of the interest. And this is, I think, from a theoretical standpoint, but also from the current situation, because we know that the inflammasome is triggered by alum. So alum is actually engaging this way by creating these kind of these crystals, yeah, which then in the inflammasome is triggering the immune activation. ATP has its own channel, which also leads to the activation of dendritic cells. Then toll-like receptor, as I showed you before, is engaged by several of the mediators, such as, I'm sorry, I have to go back one slide. So this is engaged by several adjuvants as well, and then also C5, C3a. All these mediators can be engaged and have been explored, and at the end of the day, most of them lead to the activation of MHC class II, CD80 and CD86. Why are these molecules important? Because they are critically involved in the activation of T cells. This is the first signal. The second signal is a co-stimulatory molecule which is necessary to drive the naive T cell into proliferation, into differentiation as well. And here CD80 and CD86 are instrumental. So if we now look at the adjuvants, I would like to go a little bit into detail what has been published and what do we really know about this antigen uptake. So this is, of course, the first step, right? I mean, even we could also enhance the antigen uptake. And here, what we actually learn, so if you inject a fluorescent protein, yeah, into a mouse, and then isolate the dendritic cells from the lymph node, you can see here you see the time scale 6, 24, 72 hours. That antigen uptake is actually quite rapid and quite efficiently, and also the if you look at it, the quantity, it's quite quick. Yeah, these are several animals. You see there's some variation amongst not the animals, but the CpG is a little bit faster and efficient compared to alum, and this the lower one here is PBS. So this is the diamond, yeah, which is the control. And you can see here this is for B cells, and then there's conventional dendritic cells and plasmacytoid cells. So it's not a very big difference between the different groups. Now, if we now look at the formation of the complex between antigen and MHC, so when it's displayed on the surface, so this requires antigen presentation and also the transport of the MHC molecule then to the surface of the cells. We can stain this by antibodies in certain systems here with this antibody called YAE, and you can see here again the time scale, and you can see also that there are differences between different cells. Again, the adjuvants, if you compare the adjuvants CpG or alum, yeah, they are actually enhancing the activation of the B cell and therefore also the presentation on the surface. And the same holds true actually for conventional dendritic cell. Interesting here is, however, if you look here at the phases, that there is a slight delay. So B cells and conventional cells are, I would say, similar. But if you now look at plasmacytoid cells, they are delayed. Yeah, so they are presenting the antigen later than the other two antigen-presenting cells. And these are this is a publication which I can really recommend from Hutchison and colleagues in FASEP 2012. So if we are now moving on, I think here this artist has actually lost his ear, and he did this intentional. Yes, it's Van Gogh, and the reasons why he actually removed his ear are not very clear. But I think scientists got very much motivated by Van Gogh and actually removed the injection sites. They removed the ear of mice where they injected the adjuvant. And this is kind of interesting in regards to the discussion whether you need a depot for the antigen to make it a good adjuvant. So the idea is that you need prolonged and continuous release of the antigen to generate an efficient situation of antigen stimulation, and therefore also generate a stable T cell phenotype. So the idea is really that you need a longer time period. But surprisingly, this is actually not really the case, yeah, because you can see that already after 2 hours, you already have maximal levels of antigen presentation. So, you know, removing the depot really doesn't have so much of an effect. So the antigen is very quickly transported into the draining lymph node, and whether the antigen is still in the depot or not does not seem to matter so much. Yeah. So I think you know, if this depot discussion, if you hear this somewhere, I think you should be skeptical, because there's no so much evidence for that really. Now, the other question is the antigen persistence needed? Yeah, and here's a very complicated model, and I have to ask to bear with me with this. I try to simplify it as much as you can. This is a study from our colleagues here in Munich, published from Rammstein et al. in the JI 2014, and they used a transgenic model where they were actually able to switch on and off the antigen presentation by a transgenic system where the antigen expression is actually inducible by a TET system, yeah, which is coupled to the MHC molecule. So both MHC and antigen in that sense are under control. And what these colleagues showed is that these are the days where they adoptively transferred the mouse to a recipient where the cells were expanded, and you can see here that 24 hours after a transfer, you see this nice proliferation. This is in CSFE staining. So here you see the cells which do not proliferate. Yeah. Here is the time of treatment with DOX. So in other words, the induction of the antigen. And you can see very nicely here the proliferation of the day, starting on day 4 or -4 or -1 after feeding with doxycycline for 24 hours. So this is basically showing that the system works, yeah. So you can actually switch on and off the presentation in this defined time window. And here you have two, I should mention this, they used CD4 system, and they also used the CD8 system. And now here we are looking now at the need for persistence of antigen presentation. And I think this is very interesting because it really shows that some of the genes do not need antigen persistence. Yeah? And you have here, as an example, the differentiation of Th1 cells and those cells, which those genes. Yeah? So they removed the T cells of these animals after this induction and compared this to transient expression or persistent expression. So transient where they switched off the MHC, and persistent where they left it fully wild type, so present all the time. And all the genes that are here directly on the line means that they are independent of antigen presence. And you can see that some very well known genes of Th1 cell differentiation, such as Tbet, are pretty much here on the diagonal line. Yeah? And this really shows also IL-6 receptor, IL-7 receptor. You know, these are all, on one hand, activation dependent, but they are not dependent on the persistence of the antigen. So I think this is very interesting because it really means that with the persistence of the antigen, we are not so much driving the T cell phenotypes, but we are changing other components of gene expression, which are probably these genes up here. So I think this is important to know. But I think another very important aspect in the presence and in the timing of the antigen's presence are the immunoglobulins. And this is a very old publication from 1990, so this is already very well settled knowledge, and this really shows that the B cells really need the antigen's maturation in several cycles. So, and what you can see here in this presentation, this is IgG1 and IgG4, and these are the days after vaccinations down to a year. And what you can see here is an inhibition assay, and the countings here, or the amount, the percentage of inhibition is shown here on the right side, and you can see the more the peak is going to the left side, the higher the affinity of the antibody is. Yeah, and this really shows that starting from day 14 after vaccination, you get an increasing affinity of antibodies, which is kind of increasing by time. Yeah, and now we have the secondary and tertiary vaccination. So these are kind of booster vaccinations, and yet then you can see that the affinity of the antibodies, so the maturation, really moves to the left side. So I think this illustrates very nicely that particularly the second booster is really necessary to get nice affinity of antibodies, particularly of IgG4. You can see it here very nicely. Yeah? And this may also explain why we have to wait so long until allergen-specific immunotherapy works, because there is this maturation process. But this has nothing to do with the persistence of the allergen at the time of T cell presentation. So I hope I could illustrate you that the depot should not be misunderstood as necessity to drive the T cell response, but that it is important for the B cell response. But this probably needs larger time periods to be effective. Now there comes the bad news. Alum is actually, yes, it's enhancing antigen presentation, particularly delivery by the B cells and dendritic cells. This is what I've shown you. But the bad news is that alum is actually enhancing type 2 immunity. Here you see a vaccination in a mouse, you see the proliferation, the open circles are ovalbumin vaccination alone, and then you have ova together with alum, and you see that alum is accelerating a little bit the proliferation. You see here the presence in the non-draining lymph node. And you see also there a minor difference, but I think the very important thing is here that the cytokine secretion of these cells, shown here in this Journal Experimental Medicine paper 2008, is significantly increased. You don't even see ova alone, but together with alum you see a clear boost of type 2 cytokines. So this is considered as a non-wanted side effect of the alum. And this is one of the reasons why there's considerable research which is trying to replace alum by other adjuvants. Now alum triggers also MHC2 expression co-stimulation, so I've shown you similar results in the previous studies. This is shown again by the work of Acknolwdgements here by flow cytometry. I mentioned CD86, now you can see it here nicely after 6 hours. You see an increase of CD86 expression, which then is present for prolonged time period up to 24 hours. There's a decrease here to some degree. It also enhances then CD25 expression, so this is the IL-2 receptor, which is important for Tregs, but is also generally an activation marker of T cells, and you can see this very strong activation, which is really alum dependent if you use it together with ovalbumin. So again, this really shows here that alum is one of the well-established adjuvants, which is on one hand nicely triggering the inflammasome of the dendritic cells, but on the other hand also enhances T cell activation quite nicely, but it unfortunately somehow enhances the type 2 phenotype. And now I want to come to future perspectives of allergen-specific immunotherapy, and I want to show you one approach which we engaged in our research, which I always found very exciting, and this is an option of fully degradable matrix, which is actually even FDA approved, and there's a freedom to operate. There's no patents which restrict the use. It's actually already utilized in anti-cancer drugs. And what it is, it's a polymer of lactic co-glycolic acid, PLGA, and it's combined with elements of polyethylene glycol. And the titration of these two components then determines how this polymer actually behaves. And the interesting thing is, if you can now titrate these elements, and what happens is, if you increase the temperature from this fluid state, so you now increase it, and then suddenly you get a gel state. So this is exactly the opposite of what you usually see from components. I mean, usually if you heat some sort of substance, it gets liquid in the best case, like sugar or something like that. But here it's exactly the opposite, So it actually gets from solid into a gel state. And this now can be titrated in a way that when you have it as a fluid form in your vial, and you can see it here in this image, and now you heat it up to body temperature, it gets into this gel state. So this is quite, the situation we want to have at the site of injection. So here we can now mix in immunomodulators, into this substance, and then at the site of injection, due to the body temperature, it gets into this gel state. So this is illustrated. So we mix in now drugs. And now we can take it out of the tube by a syringe, no problem, because it's still liquid. Now we inject it into the body, and then it gets into this gel state, and from the gel state it can now release the active components into the environment. So this can be the antigen, but you can also combine it with other substances. So this is, we think that this is quite useful, and it's fully biodegradable. So in contrast to alum, where we have the problem that maybe alum may also have some side effects. So this is disappearing simply. And as mentioned, depending on the titration of the components, this is an assay here where we use calcitriol, because this can be very nicely measured in the periphery of mice, and then you can see the release rates. This is an in vitro experiment here now where you can see that there's a continuous release of this substance out of the hydrogel. But we also analyzed this release with gadolinium labeled albumin, and you can see here an animal in a cross-sectional image. So we did a PET scan analysis. This is a marker, a tube which we put next to the animal when we did the imaging of the anesthetized animal, and then we had two injection sites here at the left and at the right flank, as you can see here. And we monitored the mouse 2 hours after injection and 88 days later. And you can see very nicely here the depot. Here we have a lower amount, and then you can see how this depot is actually getting degraded and simply disappears. So this is a nice way how we can actually bring immunomodulators into the animals. And I would like to remind you to our concept that actually immunotherapy will profit from a differentiation of regulatory cells, where we inhibit the commitment towards type 2 or also type 1 phenotypes. And we performed a clinical double placebo randomized clinical trial together with Steve Durham at the Imperial College at the time when I was staying there, where you used an antibody which neutralized IL-4. So this directly relates to our hypothesis I showed you in the previous slide, that we think that IL-4 is torpedizing the commitment of regulatory T cells. And then we analyzed in this study what this neutralization of IL-4 during the vaccine, during the updosing phase, would do to the patients during the season. So when, due to a seasonal impact, IL-4 is elevated. And what we did is we analyzed skin prick test and looked also at the serological parameters, and what we observed is unfortunately no effect on the skin prick test. So this was not changed by this approach, but maybe skin prick test is also not the ideal endpoint for regulatory T cells. So a clinical endpoint would have been preferable, I think. But very interesting and very impressive was the effect on the T cells. Because what we could really show, and here we see IL-4 ELISpots in the response to the specific allergen. So when you do these assays, you quantify the number of clones in the peripheral blood which are responding to the treatment allergen. And you see here the baseline. We have the same here, IL-10 ELISpots. You see here the placebo group, and here the treatment group where we had immunotherapy in the combination with anti-IL-4, and you see a significant decrease during the pollen season. This is here, and here at the clinical endpoint a year later. So you see even a year later, and we only treated during the 14 days of the updosing, not in the continuous vaccination shots. Only in the updosing you see this continuous decrease of Th2 cells. So this is really a success and shows that immune modulation at the time of vaccination has a long-lasting effect on the immune system here on these individuals which are undergoing allergen-specific immunotherapy. We did not see a difference in IL-10 ELISpots. Now, another option would be also to use JAK kinase inhibitors. And if you remember the slides, and tofacitinib is one of the JAK inhibitors which are clinically used. So, this is a licensed drug, and we filed a patent to use the small molecules in immunotherapy. And just to remind you, these JAK kinase inhibitors are actually inhibiting IL-4 signaling. It inhibits IL-6 signaling, etc., etc. Yeah? So these are actually the cytokines which we think are impacting T cell differentiation of the T cells on their way to the regulatory T cells. So what we tried in an experimental animal model of allergen-specific immunotherapy, where we immunized the mice here at day 0 and 1, and then we performed subcutaneous injection of the antigen, in that case ovalbumin, in the combination or without tofacitinib. So the idea was that we, like in the clinical trial with the graft-specific patients, which I showed you in the previous slide, here in this animal model, we wanted to cover, to protect the antigen vaccination with this JAK kinase inhibitor, sort of blocking all pro-inflammatory cytokines. And what we observed is that, first of all, here this was kind of our endpoint. This is a cell infiltration of cells into the mouse lung, and unfortunately the immunotherapy itself was already so efficient in reducing infiltration that we had no room of further improvement by the drug tofacitinib. However, if we look at the infiltrating eosinophils, which are kind of a reflection of the type 2 immunity in these animals, we can see there was a significant difference here between these two conditions. So this really illustrates, in fact, that we are able also in an experimental model to use this kinase inhibitor to block the type 2 exaggeration of which we observe in these animals. We also looked at total IgE. There's maybe also attendance, but it's not statistical significance. But interesting was, and this is also very important, I think, that the immunoglobulin production was not affected by the JAK kinase inhibitor. So this we also took this as a positive incidence, because we think that IgG4 is an important molecule in allergen-specific immunotherapy, and IgG1 maybe in the mouse kind of a paralog. I mean, you have to remember that IgG4 does not exist in mice, but at least IgG1 is not changed, and we anticipate that maybe IgG4 induction would not be affected by a regimen like that. We also looked at all kind of cytokines. IL-4 was not changed, to our surprise. We hoped that we would be able to see a difference here, but we couldn't see any. But interesting enough, the monokines, particularly IL-1, was significantly changed. Now, this may be related to the fact that we are using alum here in this model. Maybe this would not be the case if we would use another adjuvant. But also in our clinical trials in Pacific study, which we published 2 years ago, we also see that there's a general immunosuppression. And I think this is something which we want to investigate in the future. I think an interesting and yet unknown phenomenon is the role of the monokines and monocytes, dendritic cells, in immunotherapy. So they probably play a bigger role than we currently anticipate. We also see this effect, by the way, on the plasma level of the cells, which really gives us confidence that this is a relevant finding. I would like to conclude my presentation now. What we have seen is that your ear is important. Watch out for it if you are together with experimental scientists in immunotherapy. Alum-free alternatives exist, yeah, but they are not fully investigated regarding their immune differentiation capabilities. I think this is research that is very much needed and is currently lacking. I think we have to better understand the combined T-cell, B-cell and also monokine response to allergen-specific immunotherapy. Immune modulation allows long-lasting reprogramming of the immune memory. I've shown you this clinical trial. I think this is a very important and stimulating result. So we can use biologics, for example, to long-lastingly change immune memory if applied together with the vaccination. So the co-administration is key to this, and I think you need also sufficient amount. Immune suppressors may be essential to generate a non-lytic vaccination success. So I think in driving immunofunctions, we can actually switch from a lytic response such as interferon gamma to a regulatory response. This has to be the aim for future trials. Tofacitinib, I've shown you, does not inhibit T-cell differentiation, so that's, I think, interesting. And this, by the way, a difference to cyclosporine. So if you consider a different immunosuppressive drug as an adjuvant in immunotherapy, tofacitinib is, I think, useful. Cyclosporine is not. By the way, cortisone is also rather promoting regulatory T cells, which I think also supports this concept. Tofacitinib could provide an ideal immune modulator for immunotherapy and candidate for licensing this patent have indicated their interest, but I think generally speaking, and also the reason why we pretty much dropped this approach for the moment is that really large investments are required in this area, and the immunotherapy companies do not have this capacity at the moment. I would like to acknowledge outstanding researchers who worked in this area, particularly Adam Chaka, our ENT colleague in the clinic, but also Jan Gutermuth, who actually brought in the idea of tofacitinib as a co-adjuvant into the story. Also Antonio Aguilar Pimentel, Anke Kressel and Dennis Huskamp, as well as Simon Blank, one of our group leaders who's very actively involved in the experimental immunotherapy models. I do thank you very much for your attention, and I hope you like this presentation, and I would appreciate if you press the like button in this channel, and hope to see you soon again. Bye bye.